Assalamu alaikum. So in this lecture, what we will do is we'll talk about two techniques. And these techniques, they are not used anymore as extensively as before. But if you understand the concept, and that's what I care about, it will help you out understand a lot of things in molecular biology. So the techniques are known as dot blot and southern blotting, or dot blotting and southern blotting, whatever. Okay, so these two techniques are based on hybridization, the concept of hybridization, which means that we're taking a DNA from a foreign source. Okay, and we allow it to hybridize, to anneal, to bind to another strand of DNA. And the concept of hybridization is based on complementarity of DNA strands. Okay, that is, again, you have an A on one strand, you have a G, or you have a T on the other strand, you have G on one strand, you have C on the other strand, and so on. So this is what hybridization is all about. Now, so here's the idea, and that is we have a long DNA strand, and we can make a shorter DNA strand that is complementary to part of this long DNA strand. Now, when we mix them together, this short DNA strand would hybridize, anneal, bind to the other strand, to the long strand. Okay, now the question is, can imperfect hybridization take place? That is, let's say that you have a mismatch. You have a one nucleotide at one position that is different, that is not complementary to the opposite strand. Can hybridization take place? And the answer is yes, under certain conditions. The conditions include that there are enough hydrogen bonds formed between the two strands. Now, again, what does enough mean? Well, this is irrelevant for our course for now. Just enough, okay? That's one. The other thing is that hybridization can take place if we allow it to happen under certain temperatures, under certain conditions, such as temperature, for example. So if we increase the temperature when we add these two together, it may not happen. Hybridization would not happen, okay, because of this one single mismatch. But if we lower the temperature, hybridization can take place. Ionic strength is something else. We already talked about it in the previous lecture. Ionic strength. So if we increase the concentration of ions, possibly charged ions would mask the negative charges between the two strands, and that would reduce repulsion. Okay, and this would allow hybridization to take place. GC content is another factor that can control hybridization. Okay, the probability of hybridization taking place. So if there are many G's and C's that are complementary, that are present, that exist between the two strands, then yes, hybridization can take place. But if you have a lot of A's and T's, then that's a problem. So the hybridization may not take place. Of course, the reason is G and C forms the hydrogen bonds A and there are two hydrogen bonds between A and T. All right. So this short DNA strand is known as a probe. Okay. So probes are basically also known as oligonucleotides. So oligo means short. Okay, so it means something, it means it's a polymer, for example, that is formed, or that is composed of 3 to 7 subunits usually. But oligo means short. If I say oligonucleotide, it's like saying oligo. It's like a short sequence of nucleotides. So it's a nucleic acid, but it's a short nucleic acid. And we use this probe to search for a DNA sequence that is complementary to the probe. Okay, so we use this probe, this short nucleic acid, this short nucleotide, to search for the possibility of an existing region in a long DNA that is complementary to the probe itself. And usually we label probes. Remember the difference between labeling and staining. So we label a probe. So we attach like a fluorescent tag, or we make the probe radioactive using radioactive phosphorus. So wherever the probe goes, we can see it. We can observe it. We can detect it. Okay. So again, the probe does not have to be 100% complementary to that region in the long DNA strand. Okay. Of course, it's preferable that it's complementary, but it doesn't have to be 100% complementary. So whenever we add a probe to this long DNA strand, there would be competition between the long strand and its complementary strand and the probe. Okay, and because whenever we add a probe, we add it in abundant quantities. Okay, so there are many more probe molecules compared to the original DNA strand, or let's say that what we call the template. Okay, that is the copy or the DNA strand that I'm testing. And because of that, there is competition. And remember that hydrogen bonding is non-covalent, so it's reversible. Okay, so sometimes, yes, you can have hydrogen bonding between two molecules, but it's reversible. So if you add a probe, there would be competition between the probe and the DNA template. Okay, and because there are many more probe molecules compared to the template, to the original DNA strand that I'm testing, the probe would win the competition because there are many more of these probe molecules. Okay, so the probe would win, and you would have the formation of a hydrogen bonding between the probe and the complementary region of the DNA strand. Okay. Now, so here's the idea, and that is we have a DNA strand. This is the concept of dot blotting. And that is we take a DNA strand and we add it to a piece of paper, sort of paper, and we call it a membrane. Okay, so we can place it here, we can place another DNA molecule right here, a third, a fourth, and so on. And then we add the probe. Now, if the probe is complementary to a region in the DNA strand, like in here, for example, you would have this annealing or hybridization taking place between the two. Now, if the probe is not 100% complementary, and we use strict conditions, that is, we increase the temperature, we add denaturing molecules like formamide, urea, for example. If we reduce the ionic strength of the solution, if we reduce the GC content of the probe, for example. So under strict conditions, annealing may not take place. Okay, but if we make conditions light, then annealing can take place. Okay, and this is, by the way, an advantage, and I'll tell you why later on. So before going into the dot blotting technique, let me introduce a few terminologies. So this is sort of like called a pedigree. Okay, so basically it's a drawing of the distribution of a phenotype or a genotype. Okay, so a genotype is the genetic composition of a person. A phenotype is the appearance, the visual appearance or characteristic or feature on that individual, or that individual possesses or has. Okay, so here we have a family. You have mom, dad, and they have 3 kids. Okay, 3 children. Now, of course, there would be genetic transmission. That is, genes would be transmitted from both parents, or from parents, from mom and dad, to the children. Okay, now these genes would be transmitted. Okay, so let's talk about the eye color gene. Now, by the way, like 2-3 weeks ago, scientists discovered that there are 50 genes, more than 50 genes, that can control eye color. But let's say that we have a single eye color gene. That is, it's a gene that determines what eye color is. Now, these genes, or genes such as eye color genes, have alleles. Okay, so what does an allele mean? So an allele is a form of a gene, or a type of a gene. So we all have the eye color gene. Okay, and this eye color gene is located on the same chromosome, on the same location, exactly for every single one of us. So let's say that it exists on chromosome 5. Now we are diploid, our cells are diploid, meaning that we have 2 chromosomes, or 2 copies of every chromosome. So we have chromosome 5 from mom, we have chromosome 5 from dad. And genes are organized exactly on the same location, same spot on both chromosomes for all of us, all human beings. Okay, so the gene is located on chromosome 5, okay, right here and right there. Now there are different alleles for the eye color gene, or different forms of the eye color gene. So we have the blue allele, we have the brown allele, we have the black allele, we have the green allele, and so on. So there are so many variants of genes, and these variants are alleles. Okay, so let's say that mom has a blue allele, the eye color gene. She has the blue allele, the blue form, and the brown form. Dad has also the blue allele and the brown allele. Okay, so this means that both mom and dad are heterozygous. Okay, they are heterozygous, meaning that for the eye color gene, the 2 alleles are different. Okay, this means that this person is heterozygous. Now, if a person has the same allele for the eye, or alleles for the eye color gene, then the person is what we call homozygous. So homo means same, hetero means different. So both mom and dad are heterozygous for the eye color gene. Someone would say, Why are they heterozygous? And I would say because each one of them has two different alleles. They have the blue eye color gene or allele, and they have the brown allele. So they are heterozygous. Now, there will be transmission of these alleles to their children. So let's say that the blue allele is dominant. Aha. And the brown allele is recessive. So what does that mean, dominant and recessive? It means that if you have two alleles, one of them would prevail, one of them would dominate the other allele. So the other allele would, the phenotype of the other allele would not be shown. And the dominant allele would be present on that individual. It would be the phenotype. So let's say that the blue allele is dominant. So this means that if a person is heterozygous, the blue allele would win and the person would have blue eyes. Now this person is what we call homozygous because both alleles are exactly the same. This person is heterozygous because both alleles are different. And this person is homozygous. Now, this person has the eye color gene, the alleles of the eye color gene are both brown. So, the recessive phenotype would be shown on that person. And the person would have brown eye color gene. Okay? So now we know what a pedigree means: allele, dominant recessive, and homozygosity or heterozygosity. Okay. Now, so dot blotting is based on the use of a piece of paper which we call a membrane. Okay, and there are different types of membranes, but this is a scanning electron microscopic image of a membrane. Okay, so that's how it looks like. By showing these images, by the way, I'm not fooling you. This is a real image of a membrane, and that's how it looks like. Okay, so the idea here is that we add DNA to this membrane by here. And the DNA would be embedded inside the membrane. It would stick to the membrane. It would attach to the membrane. And when we add DNA, we're adding, let's say, a single type, but many molecules, by the way. So it's not just one molecule. No, many molecules of DNA. This DNA can be simple, so it can be a single type or, let's say, strand of DNA, or it can be a whole genome. Okay, what I mean by genome is a whole DNA of a human cell. Okay, so we add this DNA to the membrane, and the membrane would attach to that, and the DNA would attach to the membrane. And then we add a probe. Now, if in this whole DNA there is a sequence, okay, a region that is complementary to the probe, then the probe would bind to it. It would hybridize to it. Okay, and remember that the probe is what? It's labeled. So if attachment takes place, the spot, the place where the DNA is located or where the DNA is spotted, it would emit a signal. It gives a signal. If there is no complementary sequence in this DNA, or a sequence that is complementary to the probe, then the probe would not bind, and as a result, the spot would not emit or give us a signal. Okay, so that's the idea of a dot blot. Now, So here's an example of an application of dot blotting. And this, when we can use dot blotting to diagnose cystic fibrosis. So cystic fibrosis is a respiratory disease. It's a condition. It results in the formation of an abnormal lungs, and a person would not be able to breathe normally. So the cause of cystic fibrosis was found to be deletion of a specific gene, the cystic fibrosis gene. Okay, so normally a person would have a gene, or within the gene, normally a person would have the following sequence. Of course, it extends left and right. Okay, so it's long sequence, but this is just a region of the gene where this mutation, okay, would happen, normally or abnormally happens. So you have deletion of AGA, deletion of 3 base pair, okay, at position 508. So the triangle means deletion. 508 is the location of the deletion within the gene. So you have deletion of this, and notice that this is the 5' end, the 3' end, and notice that we're showing only one strand. We're not showing the complementary strand, okay? There's no need to. Now, individuals with cystic fibrosis, they have this deletion. So the AGA is missing. So you have everything except the AGA. Okay. So in order to detect if a person has a deletion or not, we use an allele-specific oligonucleotide. So we've already defined oligonucleotide, so short nucleic acid, a probe, in other words. And this probe is allele-specific, meaning that it detects an allele, and the allele that it's detecting is the allele with the mutation. Okay, the form of the mutated form of the gene. Okay, so there are 2 alleles, okay, that we're using. We have this allele which detects the normal allele, the normal gene, and we have the other allele that detects the allele with the deletion, the AGA deletion. Now notice that there are some parts within the probe or the oligonucleotide that detects the mutated gene that are complementary to the gene, but because we're using strict conditions when doing hybridization, this probe would not bind to the normal allele. Okay, and this one here would not bind to the mutated allele as well. Okay, so these probes, these oligonucleotides, are allele specific. Okay, so we have a pedigree right here. And the pedigree basically tells us that here we have a male, and males are represented as squares, and we have a female right here, and females are represented as circles. And there's a line between them, meaning that they are married to each other. And this line right here that is going downward means that they have children. So they have 3 children, okay? They have a female, and they have a male, they have a boy, and they have another girl. Now, a filled circle means that the person or the female is affected. She has cystic fibrosis. She has the phenotype. She has the disease. If the circle or square is not filled, it means that the person is normal. That is phenotypically normal. Okay, that is, the person doesn't have any symptoms for cystic fibrosis. Okay, so here we have, okay, so this is the another representation, nice representation of the pedigree. Now, so dot blotting was performed on this family. So DNA was taken from the father and spotted on a membrane. Same thing with DNA from the mother and the children. So right underneath each symbol, you have the DNA of that person. Okay, so DNA was spotted for the father, DNA of the father, DNA of the mother, DNA of the 3 children. Now, for this membrane right here, the normal allele specific oligonucleotide, that is the oligonucleotide, the probe that detects the normal allele, was added. And for this piece of membrane, the other allele that detects the mutation or the deletion was added. Okay, now whenever you see black, it means there is a signal because we have the probe labeled with a fluorescent tag. Okay, meaning that when it's added to the membrane, if it binds to the DNA, the spot would emit a signal. Okay, so notice here that for the father's DNA, there's a signal that is emitted from both spots, meaning that the father has a normal allele and has the allele with the deletion. So the father has 2 chromosomes, right? The father has a normal allele and on one chromosome, and on the other chromosome, the father has the allele with the deletion. So we say that the father is what? Homozygous or heterozygous? We say that father is heterozygous. Look at the mother now. Now, there is a signal that is coming out when the normal allele was added and the mutated, or the allele that detects the mutated allele. Or, yeah. Okay, well, let me say that again. So there's a signal that is coming out when the normal oligonucleotide was added and when the mutated oligonucleotide was added. This means that the mother is also heterozygous. Okay, and we call them as well a heterozygous person. We also call them, especially if we're talking about an allele that causes a disease, we say that the person is a carrier. Okay, that is, a person is not really affected. Notice that the square and circle are not filled, meaning that phenotypically both of these individuals are normal. Okay, but they are carriers. They carry the mutated allele. And this means that what? The mutated allele is dominant or recessive. It means that the allele is recessive. You need both alleles to be mutated to be presented with a phenotype. Okay, so this means that the allele is recessive. Now, let's look at the children right here. Now, this daughter that phenotypically has the disease that has cystic fibrosis, when the normal allele specific oligonucleotide was added, there was no signal. But when the other allele, the mutated allele, was added, there was a signal. This means that this individual, this little girl right here, has both alleles as the mutated alleles, the alleles with the deletion. And that's why she has the disease, or she is presented with the disease. Okay, because she doesn't have the normal allele. Let's look at this boy right here. The boy has signal coming out from the spot when the normal allele specific nucleotide was added, but not when the mutated allele was added. This means that this person is homozygous, just like this person, right? That's just like his sister, except that he has both alleles as the normal alleles, and he doesn't have the mutated allele, whereas his sister has both alleles with the mutation, with the deletion. She doesn't have the normal allele. Let's look at this third sister right here. She has signal coming out when the normal allele specific nucleotide was added and the mutated probe was added, and this means that this person is a carrier. She is heterozygous. Okay, so this is dot blotting. This is how we can use dot blotting. So simply we say that the father and the mother are both carriers, and they have, let's say, these children right here. This boy is not affected. Okay, this girl has cystic fibrosis, and these two are both carriers. They are heterozygous. Okay, so I think this is the link. All right, so let's talk about southern blotting right now. Now, this technique is really a combination of DNA gel electrophoresis and hybridization. So in dot blotting, all we did is just hybridization, okay? But for southern blotting, we do both. We do electrophoresis followed by hybridization. And it's called southern blotting because the person who invented it, his name is something southern. Okay, so they called it southern blotting. Okay, so what southern blotting would allow us to do is to detect the presence of a DNA segment that is complementary to a probe, just like dot blotting. And number two, we can determine the size of the DNA fragment. Now, how do we do that? Well, we do electrophoresis followed by dot blotting, and that's how we do it. So DNA is taken from an individual, and this DNA is fragmented by restriction endonucleases. Okay, we will talk about restriction endonucleases in the next lecture. So the DNA is fragmented. So you have the production of DNA fragments from this whole genomic DNA. So genome is basically the total collection of DNA in a cell, or total collection of DNA in an individual. Okay, so we say the human genome, your genome, or we can say the cell's genome. So the DNA, the whole genomic DNA, is fragmented into smaller pieces. Then these fragments are separated using agarose gel electrophoresis. Remember, we talked about that in the first lecture of this series. So the DNA fragments are separated according to size. So here we have the well, and the DNA is moving, DNA fragments are moving towards the other end, and they are separated according to what? To size, okay? To length, in other words. Okay, that's what we do. Then we take the gel. This is the gel. That's how it looks like. These are the wells. And here we have sample 1, sample 2, sample 3, sample 4. And each sample has DNA fragments of different sizes or different lengths. Since we're starting from here, here we have the large DNA fragments right here, and here we have the small DNA fragments right here. So these are the largest DNA fragments, and these are the smallest DNA fragments, and here we have everything in between. Okay, so then the DNA is placed on top of a membrane, or actually underneath, but it doesn't matter. Okay, so it's placed underneath a membrane, and these DNA fragments are transferred from the gel to the membrane. Okay, so you have the membrane right here on top. So the DNA fragments move upward onto the membrane, and and the DNA fragments are now attached to the membrane. Okay, now notice something, that is, the location of the DNA fragments is exactly on the membrane as they look like on the gel. So you have these two, for example, right here on top. You have them also on top. You have, let's say, these two on top, and you have them also on top right here. You have the small ones right here. So basically, you have the small ones here, and you have the small ones also right here. So basically, the membrane looks like a replica of the gel. So you have the DNA fragments also organized according to size or length. So you have the large ones here, and you have the small ones right here, and everything in between. Okay, then we take this membrane and we add a probe right here. Now, if there is a sequence within these DNA fragments that is complementary to the probe, what would happen is that the probe would bind to the DNA strands, DNA fragments on the membrane. Okay, so you'll have the probe bound here, here, and here, but not to the other DNA fragments, because they do not contain a sequence that is complementary to the probe. Great. Then we wash away all of the non-bound DNA. Okay, and what happens is that, okay, my picture is covering this, but it doesn't matter. Okay, so you get the idea, I hope. So, and then the membrane, the fragments that have the probe bound to them would emit a signal. So we would have a signal coming out from here, 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 and here, and so I know that these samples, for example, these samples have a DNA fragment that has a sequence that is complementary to the probe. That's one. And number two, we know what the sizes of the DNA fragments are. Okay, so we know two things. One, if a DNA fragment that is complementary to the probe exists in the sample. And number two, we know the size of the DNA fragment, because the DNA fragment migrates through the gel and according to size, and it's located on the membrane exactly like how it looks like on the gel. So we know what the sizes of the fragments that have a complementary sequence to the probe are. Okay, so these are the two things that we know. Okay, now pay attention to something. That is, the size of the fragment, or the sizes of the fragment, do not reflect the size of the oligonucleotide or probe. Okay, so the sizes of the fragments do not reflect the size of the probe. They reflect their own sizes. Okay, so here, for example, let's say, again, that's the last example. Let's say that we have two samples, sample 1 and sample 2. Okay, and let's say that they have fragments of different sizes, and these fragments have complementary sequences to the probe. So the probe right here would bind to a large fragment and would in one sample, in sample 2, and would bind to the small fragment in sample 1. So in the gel electrophoresis, when we when we separate fragments according to size, we have a lot of fragments, and we will not be able to distinguish these fragments from each other. Okay, according to sequence, we wouldn't know the sequence of all of these fragments. Okay, but then when we transfer them to a membrane and we add the probe to them, to the membrane, now the probe would bind to these two fragments, and a signal would be emitted. And again, so I know that samples 1 and 2 have DNA fragments that are complementary to the probe. That's one. And we know what the sizes of the DNA fragments are because that's how they travel in the gel electrophoresis. Now also pay attention to something that is really important when we present a gel. When we show a gel, we show it in a way whereby the large DNA fragments are shown on top and the small DNA fragments are shown in the bottom. Now notice something, that is, the sizes that are shown on the membrane reflect the sizes of the fragments again, not the probe. Allah ma'akum.